Hello, welcome. This is Leah Adams and Ron Coleman. Today I'll be presenting Exploring Fidalgo Bay's Forage Fish, a decade of egg-centric sampling. In this presentation, we will cover the abundance of forage fish eggs that are being found on the Fidalgo Bay Aquatic Reserve, as well as go over the data and see what it can tell us. This presentation will cover all the sampling that took place on the Fidalgo Bay Aquatic Reserve. What is the Fidalgo Bay Aquatic Reserve, you may be asking? So, in 2008, the Washington State Department of Natural Resources designated approximately 780 acres within Fidalgo Bay in northwest Skagit County as an aquatic reserve. As you can see on the right, there is a photo of the bay. The green highlighted section is all of the lands that are incorporated into the aquatic reserve. At this aquatic reserve, we have a very dedicated citizen stewardship committee that has been doing sampling work there for the last decade. It is because of that sampling efforts that we are able to do this presentation today.
All right, so first section, eggs, eggs, and more eggs. In this section, we dive into what, when, and where we're finding eggs and how those numbers have changed throughout the years.
This presentation is made up of 3 key points that I want you to remember over the course of the presentation and take away from all this. So the first one is, of all of the Department of Natural Resources aquatic reserves, Fidalgo Bay Aquatic Reserve has the most prolific spawning of surf smelt forage fish. As the years passed, surveys became more accurate and the number of eggs found increased. Prolific spawning at Fidalgo Bay. Distribution of egg counts for each aquatic reserve 2013 to 2023. Before we look at the graphs, I want to draw your attention to the legend at the bottom of the screen. This gives the acronyms for our various aquatic reserves, with CI being Cypress Island, CP being Cherry Point, Fidalgo Bay being FB, and so on and so forth. And also note, spawnless samples have been excluded from these graphs. And just as a note, spawnless samples are any samples that had egg counts of 0. So if you see any data points that seem to be at 0, they're not actually at 0, they're at 1, 2, or 3. Very low, but not quite 0. So we'll start by looking at the graph on the left, and you can see this is a jitter plot, and so each dot represents a survey and the number of eggs found in that survey. We can see that a lot of the data points are accumulating near that 0 and also accumulating at that 100 marker. And accumulation at that 0 is to be expected. Of course, we find a lot of eggs with very little counts, but it seems a little weird that we have a lot of egg counts right at that 100 marker, almost a perfect line, especially for Cherry Point. But this is because during our lab processing, our protocol is to only count up to 100 eggs, because some samples have thousands and thousands of eggs in them, especially if those samples are from Fidalgo Bay. Some people will count more than 100, even up to 150, because sometimes you lose track of your counts and you realize you've counted way more than you should have. But either way, that's why there's an accumulation of egg counts at that 100 range. So we'll start by looking at Fidalgo Bay. Fidalgo Bay seems to have the most data points of any of the reserves in this graph, and that's not because more surveys are being taken at Fidalgo Bay. That's because there's a lot less spawnless samples for Fidalgo Bay. Most of the time when we have samples from Fidalgo Bay, they will be samples with eggs in them. Unlike Cherry Point, Murray Island, Nisqually Reach, Production, Smith, and Cypress Island, all those places, they have less data points, not because we're surveying less, but because more of those samples that are being collected are spawnless. And since we included spawnless, those data points do not show up on this graph. So Fidalgo Bay seems to have quite a lot of surveys that have a number of egg counts at that 100 range up to 150. We still have a large accumulation between 0 and 50 eggs, but a lot of them seem to be at that 100. Then we move on to Maury Island, Nisqually Reach, and Cherry Point, and we still have a lot of eggs at that 100 count limit, but we also have a lot of samples down at that near 0, with 1, 2, 3, all the way up to 10 eggs. And then we move over to Protection Island, Smith and Minor, and Cypress Island. And we have a lot less samples that have eggs in them for these reserves. And if we do find eggs, those egg counts are very low. So for Cypress Island, we've only ever had 3 samples that had eggs from that reserve. And when we did find those eggs, there were only like 1, 2, or 3. There was not any abundance of eggs. So we turn this jitter plot into a box plot, and if anyone knows how to read a box plot, it's otherwise called a box and whisker plot. And as you can see, we have the box with this bold line in the middle being the median. For Fidalgo Bay, the bold line is right at the top of the box. But if you look at Cypress Island, Maury, and Nisqually Reach, you can kind of see the box itself with that median kind of placed more in the middle of it than on the edge, right on the edge. The lines up below the box are called the whiskers of our box and whisker plot. And so we can separate a box plot into 4 quartiles. 25% of the data is the top whisker. Another 25% of the data is between the median and the top of the box. Another 25% of that data is between the median and the bottom of the box. And the last 25% of that data is from the bottom of the box to the end of the lower whisker. So for Fidalgo Bay, we have a pretty high median with 100 eggs being counted in those samples and near the median. 50% of samples above the median have egg counts from 100 to 150, and the other 50% is from 100 to 0. And that second quartile, the bottom bit of the box, 25% of samples have egg counts between 100 and around 25. So it seems to me that if we find a sample at a reserve and we find eggs in it, we are likely to have a sample that has 100 eggs or more, even a 50% chance if that sample isn't spawnless. We compare that to Nisqually Reach. They have a pretty low median, with that median being around 20 eggs, with 50% of the data being below that amount, so 50% of the samples having egg counts below 20, and the other 50% of the data having egg counts between 20 and around 130. With the third quartile, the section of the box above the median all the way to the top of the box, that's 25% of the data being between 20 eggs and around 80 eggs. So even though the boxes between the Fidalgo Bay and Nisqually Reach are in similar locations, there's a great difference in those medians and where they're placed. And because of those difference in median placements, we can see that the distribution of eggs is widely different, even if they have similar structures in their box plots. And we can compare that to Maury Island, Cherry Point, which also have those low medians with those larger boxes that don't go as quite high as Fidalgo Bay in their range. And then we can, with Protection Island, Smith and Minor, and Cypress Island, which barely have boxes at all because most of the samples are at that 0 marker. And so from these graphs, we can say that of all of our reserves, Fidalgo Bay has the most prolific spawning of forage fish. We find, if we're going to find eggs at a reserve, it's going to be at Fidalgo Bay, and if we have a sample with eggs, we're going to find a lot of eggs. So this is why we are covering forage fish eggs at Fidalgo Bay. There's just so much data to look at and so much information to pull out of the surveys. So let's get into more of that data. What forage fish are we finding the most of? On the graphs on the previous slide, we looked at egg counts of all of the forage fish species combined and compared them between the reserves. Now we are going to compare egg counts between the species themselves. Our 3 species are surf smelt, sand lance, and rock sole. These graphs, unlike the previous one, do include spawnless samples. So if it looks like a data point is at that 0 marker, it likely is. And that means that these graphs show every survey that we've ever taken across the last 12 years of sampling. So if we look at surf smelt egg counts to start at the very top, we can see that a lot of samples have surf smelt eggs being found in them. It seems like if we find eggs at all, they're going to be surf smelt. They're pretty prolific. Compared to sand lance and rock sole, where these graphs include every sample we've ever taken at these any of our reserves, but because we're not finding sand lands or rock soil very often, we have that nice thick line of data points right at that 0. Occasionally we find sand lands and rock soil, but it's not at the level of surf smelt, as you can see. There's a lot more data points above that 0 marker on the surf smelt graph compared to the sand lands and rock soil graph, which means that if we are going to find eggs at all in any of our samples, it is likely going to be surf smelt. And that's not necessarily because sand lands, rock soil are having issues with their population or endangered or anything like that. It's because our survey is tailored to surf smelt. We sample elevations that surf smelt eggs wash up at. We are really looking for surf smelt eggs when we sample. Sometimes we find sand lands and rock soil and they wash into that elevation, but it's rarer. So what that means is that for the rest of this presentation, we'll be covering surf smelt egg counts in all of our graphs. Sand lands and rock soil egg counts have been excluded.
Let's get into more information about what surf smelt actually is. And so its a scientific name is Hypomesis proteaceus. And they're beach spawning forage fish that can grow up to 12 inches in length and live up to 5 years of age. Surf smelt are what we consider forage fish, a nutrient-rich food for larger fish, seabird, and marine mammals. Fertilized surf smelt eggs are buried in the beach by waves after a spawning event. So that's why we do a lot of our surveys on the beach itself, as opposed to like going into the water and trying to, like we do for herring eggs, and try to find where they've laid their eggs on the vegetation. They will wash up very nicely on the beach, and then we can scoop sand up into our, we call them sand babies, and take them back to the lab.
Where and when are we finding the most eggs? Let's look at egg counts between the east and the west sides of the Fidalgo Bay Aquatic Reserve. The east is represented by the green boxes in our box plot, and the west is represented by the yellow boxes in our box plot. And our plot describes data collected between the years of 2013 and 2023. And just as a reminder, spawnless samples are excluded from this graph, as well as any egg that is not a surf smelt egg. Okay. Let's start by talking about a change in process. So in July 2015, we started blue bulling our sand babies, which was a method to remove the eggs from our sand babies into a smaller container that was easier to process. Before blue bulling, we used to use a method called the winnowing method, and a lot of eggs were lost during this method, so it seemed to be a little less accurate than it could have been. In 2015, we started a process called blue bulling, which allows us to preserve 99% of eggs in any given sand baby and take those eggs to the lab. And so after that, we got a lot more eggs generally in any of our samples, and those egg counts were a lot more accurate than they had been before. Okay. Let's look at the years 2013 to 2018. We can see that the medians are low for both sides. Most egg counts are lower 0. You can see that bold median is near the bottom of the box of our box plots for any of those years. That means we're really not finding that many eggs. We're still finding eggs, but 50% of our data is at that median or below, so are at the lower side. From 2019 to 2023, medians increased for the east side. Most egg counts are very high on that east side. For the west side, we still kind of are getting that lower median compared to the east side, but it is definitely a lot higher than it was in 2013 to 2018. From some artifact of changing in surveys, becoming more accurate, over the years we became a lot better at locating eggs and getting them into sample jars and to the lab for processing. For the east side, you can see that our medians are almost always at the very top of that box. All the way up in 2023, it looks like that almost 100% of our samples were at that 100 marker. There was very little samples at all that had anything less than that. 3, it seems like, if you look at those 3 outliers. So something about over the years, more skill in processing allowed us to collect more eggs than we ever had before. And generally, it just looks like that the east side has a lot more eggs than the west side does, which is interesting.
All right, so we're going to go ahead and start moving away from the graphs to give you guys some of the maps to look at, kind of visually look at the distribution of eggs and where and when we're finding eggs. For this first map we have here, this is like a technical like heat map, kind of showing the spawning density across the bay or the area of the bay that most of the sampling is done in. To kind of align your orientation, the darker red and the yellow area indicate more eggs found in those areas. So one note, just like a lot of Leah's graphs, spawnless samples were omitted in these maps as well to make them a little easier to read. So you kind of just see when we find eggs, here's kind of the density across the bay. As you see from the map, more eggs were found in the spit area, the east side, and then just north of the mansion, which is over here on the west side, kind of right above the gap that we don't do a lot of sampling in. So kind of besides the densest area on the spit, that yellow area right there, it doesn't really parse out much about the other areas, which are pretty uniform pink, like a dark pink. But the next map will kind of like dig into a little bit of that. Okay, so this map clusters all of the sites to help us kind of make sense of the area a little better. So all of the samples, once again, are including spawnless samples. So here the number of samples taken is represented by the size of the circle, plus the numbers inside the circle. So you can kind of see over on the far southeast, that's kind of the largest circle at like 266. So overall, between 2013 and 2023, you guys collected and processed 1,778 samples, which is amazing. So as you can see, over 200 samples were collected from the spit. So it's kind of that dark blue area on the kind of the west side there. And then from 2 locations on the east side and from a cluster of sites just below the mansion. This kind of like parses out a little bit of what you saw in the heat map, but kind of shows a little better where we actually find most of the eggs across the bay. More interestingly, the number of eggs found per cluster are represented by color. So the darker blue color indicates higher egg counts. The total eggs processed over the past 10 years is almost 77,000 eggs, with 1,000 more eggs found on the east side. Most eggs were found in the central part of the east side, with the spit coming in slightly lower.
Now that we've looked at the number of eggs collected between all the years that we've surveyed, let's look at when in a given year are we finding the most eggs. So we have 2 graphs here. The left describes the number of eggs collected over the given months of a year, and the right describes the number of samples collected over a given year. And these both are split between the east and the west sides of the reserve. So to start, let's look at the graph on the right. And we can see that there are a larger number of surveys being performed in the summer months than in the winter months, where there's less surveys being performed. This could be because of many reasons. Most people like going out on the beach in the summer months when it's nice out and warm than in the winter months when it's cold and dreary. Also, the winter time is peak holiday time, so a lot of our volunteers may not have been able to go out and perform surveys because of family obligations. If eggs were being laid evenly across all months in a year, we'd expect that the graph on the left, which describes the number of eggs collected in the samples in those months, would be similar to the number of samples collected. Those humps would be very similar. But, as we can see on the left, there are a lot more eggs being collected in the summer months than in the winter months. A lot more that can be explained by the increase of surveys being performed in the summer months. Therefore, the increase in surveys does not explain the large increase in eggs in the summer months. So in summary, most surf snail eggs are being laid in the summer months. Less eggs are being laid in the winter. This describes the spawning habits of Fidalgo Bay surf smelt. Surf smelt at Fidalgo Bay seem like they preferentially lay their eggs in those summer months. Let's move on to key point number 2. The changes in survivability of surf smelt eggs over the last 10 years does not show a clear trend. Egg survivability is influenced by seasonal changes and survey location. Egg survivability metrics. We'll use 2 metrics to measure how well eggs are surviving on the beach. Our first metric is live egg proportions. For this survivability metric, we include any egg that we found alive in our sample of any development stage. For this metric, we assume that any of these eggs that we collected would have survived to the end of their development and hatched and grown into a full-sized fish. Compared to our second metric, which is late-stage egg proportions, which only includes eggs that are about to hatch, if we had left these eggs another few days, they almost certainly would have hatched. When looking at live eggs only, we cannot be sure that the proportion of eggs would have survived. Likely, many of those eggs would have died before hatching. But for late-stage egg proportions, we can be pretty confident that those fish would have completed development and hatched and grown into a full-size fish. Therefore, late stage is a better metric for survivability than live eggs because of our higher confidence level in their ability to reach the end of development. Now that we've looked at our 2 survivability metrics, let's graph them out. So when are we finding the most amount of live eggs? So this graph describes a distribution of proportion of live eggs in the sample across all of our years. So unlike our previous graphs, which describe absolute counts of eggs in any given sample, this describes the percentage of live eggs in the sample. So that is the amount of live eggs over how many dead eggs is in the sample. So for example, you can see in 2013, the median is around 20. So therefore, that median describes a sample that has 20% live eggs and 80% dead eggs. Before we get into comparing the years, I will say again that blue bulling did start in 2015, so that our most accurate data represents the years after blue bulling started. Before then, we were losing a lot of eggs, and so we have less accurate proportions. So a lot of these have very low medians, like 2023, 2014, 2017, 2018 are also pretty low. It seems like we have a pretty sporadic distribution of live eggs across the years. There's no clear trend of increase or decrease that would describe a change in population. It seems to be completely random, according to this graphical representation. 2020 has a pretty high number of samples with high proportions of live eggs. And the reason is because we stopped sampling in the summer months after the COVID-19 pandemic began. And as we'll see later, in the summer months, eggs have a harder time surviving. So if you're surveying mostly in the winter months without surveying in the summer months, you're going to have an artificially high amount of live eggs in your sample compared to dead eggs, which is why the median for 2020 is so high. So as you can see, because of sampling differences between the years, that can affect the distribution of live eggs in your sample by a lot. So we're not seeing any clear trend in the proportion of live eggs in the sample in the last 10 years. Distributions are widely variable. Now that we've looked at metric number 1, let's move on to metric number 2, changes in late stage proportions over all the years. So like the graph before, this will describe the percentage of eggs in a sample, but instead of live eggs, we're looking at late stage eggs. And late stage eggs, as a reminder, are nearing the end of their development. They're probably only a few days from hatching. And we can be sure that these eggs would have likely survived to hatching. So this is our more accurate survivability metric. And as you can see, a lot of these box plots have very low medians. And the box plots generally consist of data lower than the one previously. The percentages of late stage eggs in a sample are smaller than the percentage of live eggs in the sample. And that doesn't mean that they're not surviving until the end of their development. It just means that naturally, because we're having a more restrictive metric, we're going to have lower percentages because we're not including all of the stages of eggs in a given sample. So don't look at the smaller percentages and think that there's eggs aren't surviving. It's just the way that we are looking at this, they would naturally have smaller amounts of percentages. Okay, so let's compare the years. So 2013 to 2023, all those medians are again near that 0 line. And this also does not include spawnless samples as per usual. So those medians are not necessarily at 0, they're like at 1 or 2. So a lot of these samples across the years only have maybe up to like 5% of late stage eggs in their sample. And that number doesn't really seem to be changing. The only year that seems to have a very high median is 2020, where over 50% of the samples has over 20 late stage eggs. And that, as I described previously, is likely because we didn't survey for eggs in the months where this mortality is the highest, in those summer months when it's hot and those eggs burn and crisp and die. We only surveyed in the winter months, so we have an artificially inflated median. If we had surveyed in the summer months, I would anticipate that we would have similar box plots to 2019, 2021, and 2022. So like before, like previously, we can see that there doesn't seem to be a clear trend in the changes of percentage of late stage eggs in any given sample. We're not seeing an increase in the amount of late stage eggs we're finding, but we're also not seeing a decrease. It seems to me that we are finding about equal amounts of late stage eggs throughout the years. Now that we've evaluated the survivability of our two metrics across all the years, we're going to separate that in between the east and the west sides of the reserve. So this graph describes the first metric for live eggs, the percentage of live eggs in a sample over the years, separated between the east side and the west side. So the east is represented in the green box plot, and the west is represented by an orange box plot. And as we can see, medians seem to be generally higher for the west side than on the east side. Those differences are not necessarily super stark. For example, in 2023, 2019, and 2015, those medians are pretty close together. The differences isn't so great, but for some years it is, like 2020, 2016, and 2022. In those years, the west has a much higher percentage of live eggs being found in those samples than on the east side. And this indicates that there's something about the west side that is allowing the eggs to survive and have a higher chance of surviving. And we will go over those differences between the east and the west side in further sections when we talk about environmental differences between the sides of the reserve. But in summary, generally the west side has a higher portions of live eggs being found than on the east.
All right, so we're going to shift gears a little bit again and go back to looking at some of the maps of the bay. So these maps show the number of live eggs found in all of the samples. Once again, spawnless samples have been excluded to kind of make it a little easier to read. As a reminder, live eggs are not the best indicator of survivability since they're only kind of a snapshot in time and they span all stages of development. But they do provide some good information on relative mortality across the bay, so we'll kind of get some information from them. The left map shows the percentage of live eggs in each sample, with yellow being the lowest percentage of live eggs and red being near 100% of the samples containing live eggs. So as you can kind of see, the east side has a lower proportion of live eggs than the west side. The east sides are mostly yellows, where the west side has some reds and oranges in them. But most of the live eggs found in samples are near the spit here. And interestingly, in the northwest cluster, live eggs occupied an intermediate percentage in these samples. So then, looking over to the right map, it clusters the same data, kind of like one of the prior maps do, kind of clusters them, displaying the total number of live eggs processed. So again, the spit comes out the clear winner here, with the northwest cluster close behind. You kind of see the numbers there. One caveat, due to the subsampling methods, the total number of live eggs, and all eggs for that matter, are much higher. That's why percentages are much more helpful in comparing when we're looking at these different things. All right, so like Leah discussed, late-stage eggs may be the best metric of survivability in the data we collect because they're eggs that have survived so far to near hatching. So they've gone through all environmental conditions, the sun exposure, things like that, and they're actually like close to hatching. So that is a really good metric, better than just live eggs. So these maps show the number of late-stage eggs found in all samples, once again excluding spawnless. The left map shows the percentage of late-stage eggs in each samples, with yellow being the lowest percent and red being near 100% of the sample containing these late-stage ones. The right map clusters the samples within an area and then shows the total number of live eggs in that cluster. So survivorship, as you can see, drops off quite a bit using this metric. So you can kind of see no matter where in the bay, like we're back down to kind of yellows and oranges, not a ton of dark reds. And you can see from the left map that the areas conducive to late stage survivability has definitely shrunk. With a few places near the spit and a few interspersed in the northwest and southwest sites. The cluster maps show kind of those same exact trends.
Now we'll look at how seasonality affects egg survivability. I talked about this earlier on previous graphs, describing how in 2020 there was a higher amount of eggs surviving than in other years because of our lack of sampling in the summer. And now we'll go into the reason why that is. So this graph describes the distribution of live eggs throughout a year. We're looking at the percentage of live eggs in a sample. So we're looking at metric 1, and this is over all of the months of the year. And to start, we'll look at the beginning of the graph. We can see that in January, February, March, and April, in those like for winter months, we have a pretty high amount of live eggs being found in samples. And we're seeing that number slowly decrease as we enter the summer months, where we have very low medians and the range of our box plots are much lower on the scale. And then as we move back into the winter months, we're increasing again. We weren't seeing clear trends like this when we looked at the differences of percentages throughout the years, but we are seeing a very clear trend as we look at the differences between the months. And so in summary, we can see that the higher proportions of live eggs are being found in the winter versus the summer. In the summer, we're finding a lot more dead eggs, and in the winter, we're finding a lot of live eggs. And that is because the eggs have a harder time surviving in the summer months when it's hot out. The sun beats down those eggs and they dry out, they mold, they are not able to make it to the end of their life stage. But in the winter months, when it's colder out, they are much more able to reach the end of their development because they're at a temperature range that allows them to continue their development. And you can compare this to earlier when we talked about when eggs are being laid in the reserve. It seemed like a lot of those eggs were being laid in the summer months where the survivability is the worst, and there's not a lot of eggs being laid in the winter months when survivability is the highest. It almost makes you want to go and tell those fish, maybe you should think about laying eggs in the winter versus in the summer so you can have a higher chance of survival. But unfortunately, we can't talk to fish. So in summary, higher portions of live eggs are being found in the winter versus in the summer.
Okay, back to some maps. So these maps split up the sites among the 3 seasons of survivability that Leah just discussed. So they are displaying the same range of live eggs from low percentages shown in yellow to high percentages of survivability shown in red. The 3 panels are kind of, like I said, split up on those groupings that Leah had. So January, February, and March on the left, and then the end of the year on the right, with the summer months kind of spread in between. So spawnless samples were once again omitted in these maps. In general, January through March showed high survivability among most sites, with obviously with the summer months exhibiting higher mortality everywhere. except the spit area. You can kind of see that's where a lot of the clustering of these like orange, maybe not so many reds, but oranges are during the summer months. September to December saw an uptick in survivability among most northwest sites. So they start doing really well, kind of over in that northwest area with a lot of reds. And then you see the percentage of live eggs still is pretty site dependent in other areas where you can kind of see yellows dispersed among reds and oranges. So that kind of gives you a good spatial visual for where these eggs are surviving, but still kind of following that same pattern Leah discussed.
How does seasonality affect egg survivability? We'll be looking at metric 2 this time regarding late stage eggs, eggs that have almost reached the end of their development. This graph is similar to the previous one. In the winter months, we seem to have this higher survivability in January, February, March. As we move into the summer months, our survivability decreases to almost 0. It cannot be 0 because, again, we excluded spawnless samples from this graph. And then as we move into the winter months, we have an increasing survivability again. This matches our earlier graph describing live egg proportions with metric 1. So combined, both of those graphs seem to indicate that those summer months have that really low survivability, likely because of the heat stress that the sun puts on the eggs. And in the winter months, there's a higher survivability because of the more acceptable temperature range that the eggs are experiencing.
All right, so similar to previous maps, the 3 sites were split up among the same 3 seasons. Instead using late stage eggs as the measure of survivability. So from the middle map, we'll start there. You can see there's very few samples that have any late stage eggs from April through August. And then survivability starts increasing in the fall months, as you can see some more red once again up in that northwest portion. And then it carries on into the winter where you start seeing these later stage eggs showing up at higher proportions on different sides of the bay. So kind of that same trend Leah's talking about.
For the final section of our presentation, let's talk about environmental effects. In this section, we will cover how shading, beach substrate, and the upland environment may influence egg survivability. Our final point, key point 3, covers how environmental effects, such as how much of a beach is shaded and the percentage of uplands impacted by humans, have a statistically significant effect on egg survivability. How does shading affect the proportion of dead eggs in a sample? I'm going to need you to switch your brains around a little bit for this one. Instead of just looking at the percentage of live eggs or late stage eggs in a sample, this time we're looking at the percentage of dead eggs in a sample. And unlike live eggs and late stage eggs, where if we had a high percentage in our graph, that would be a good thing because it means that we're having a lot of eggs that are surviving. Having a high percentage of dead eggs in a sample is not that great because it means that we're finding a large amount of dead eggs in our samples, which means that survivability is pretty low under that category. So we have a few different shading categories. We have fully exposed, 25% shaded, 50% shaded, 75% shaded, and fully shaded. Fully exposed is a beach that has no shading, is fully exposed to the sunlight. We're going to increase our percentage of shading over the beach until we get to fully shaded, which means that the beach is 100% covered with shade. There's a 100% tree cover over that beach. Let's look at fully shaded real quick before we look at the other sections. We have a really low sample size, less than 30 for fully shaded. So our box plot is less representative of the real life scenario than the other categories. The statistical magic number is around 30 for your sample size. It's pretty low sample size, but that's the number you need to have at least to start having a representative sample. And because our number is less than that, we can't say that our samples have any conclusive information about the real life scenario and how shading affects egg survivability under a fully shaded beach. And so if we wanted to start talking about survivability under fully shaded beaches, we are going to have to collect more samples under fully shaded beaches. Okay, let's look at fully exposed beaches. We have a really high percentage of dead eggs under fully exposed beaches. The median is around 80%, so 50% of the data is above that. So 50% of samples collected under fully exposed beaches have percentages of dead eggs between 80% and 100%. That is a really high amount of dead eggs in those samples. As we move on to 25% shaded and 50% shaded and 75% shaded, our medians decrease. Our box plots cover a larger range than they did under the fully exposed. This seems to indicate that as we become more shaded, we're finding lower amounts of dead eggs in our samples, and we're finding a lot more live eggs in our sample because they're reversely proportional. And then I ran a little statistics test because graphs can only tell you so much. So I ran a one-way ANOVA ranks test, which compares the medians between our different categories and indicates whether the differences between those medians is statistically significant. In statistics, our magic p-value is 0.05. So if you have a p-value less than 0.05, that means that your differences between the means is not likely due to chaos, to pure randomness. Because our p-value is 0.0001543, that is a significant value. And we can say that the differences between the means here are not just due to randomness. There is some environmental characteristic that is affecting the egg survivability in a positive way. So that test only really tells you that there is a significant difference between some of the means. We want to know which of the means have that significant differences. So I ran a Dunn's test of multiple comparisons using rank sums. And so we can see that there's statistically significant differences between the means for the fully exposed and 50% and fully exposed and 75%, which tells us that their shading is affecting the egg survivability in a significant way. For fully exposed, we have a high percentage of dead eggs with 50% and 75%. We have a lower percentage of dead eggs, and that difference is statistically significant and is likely due to the difference in shading between those samples. So, in summary, shading seems to have a statistically significant effect on egg mortality, but we can't be sure that relation is direct or if some other factor is in play. We just know that that effect is present.
All right, so this is a breakdown of the shading categories recorded across Fidalgo Bay sites. So they're clustered according to each site's vicinity to each other, sort of similar to other ones of these clustered maps you've seen before. So the size of the circle and the corresponding number inside it indicates the number of late stage eggs collected among sites in each cluster. So this was, remember, this was our best metric of survivability that we've kind of established in the past. So like discussed before, the Spit and the Northwest Beach had the highest survivability across the bay. In this case, though, the colors indicate something different. So they're actually indicating the most frequently recorded categories within that cluster. And then the Spit and Northwest, you can kind of see the two that we just showed. They exhibit 75% shaded and 25% shaded, respectively. So 75% is down kind of at that Spit, and then 25% shaded is sort of like the major category up there in that kind of northwest portion. An interesting note is the Karsten site. It exhibits about 50% shading, but the low number of late stage eggs is probably due to the relatively fewer number of samples that were taken. So that was about 124 samples taken, and then that kind of ended up being fewer eggs in general. Karsten was added as like a special site later on in the the decade of gel sampling. So over the entire like we're getting numbers from the entire 10 years over that entire time. Obviously, that one's going to have fewer samples just overall. Finally, this chart right here shows the number of late stage eggs broken down by the side of the bay and shading. So you look over on the y-axis, the east side is here on top, so it goes east side to west side. And then on the other axis, the most shaded is on the right. So these are kind of the breakdown of the category, where exposed is kind of to the left and more shaded is to the right. So you can kind of see most of the shading occurs, it kind of breaks it down where the shading is found across the bay. Most of the shading occurs on the west side of the bay, with very little fully shaded areas anywhere. You can kind of see when you get far to the right there, they're either like really light blue or almost non-existent at all. So kind of reflecting that same thing that Leah was talking about, where we don't find a ton of shade and therefore a lot of the survivability.
Okay, so we're moving on from shading to impacted uplands. So does the human impacted uplands affect egg mortality? And so human impacted uplands would be riprap. It would be if someone put like a retaining wall on the beach, or if they added sand to the beach. So just anything that is human on that beach would be considered a human impact, versus like a natural shoreline that just has trees and bushes and just natural features. Okay, and so we ran another statistical test, same one, so one-way ANOVA with ranks test and the dense test of multiple comparisons. And the one-way ANOVA said that there was a significant differences between the medians in this box plot, with a really low p-value of 9.869 to the -7th power. And if you guys know your powers, you'll know that's a very, very small number. And the dense test of multiple comparisons told us which medians had that still significant differences. And we actually have 4 significant differences in this graph, which is really good to see. We have some significant difference between natural and 75%, between natural and 100%, between 50% and 75%, and 50% and 100%. Generally, the medians between natural 25% impacted and 50% impacted seem to be pretty similar, with percentage of dead eggs in any given sample about around 50%, and just the distribution above and below being pretty even. Once we get to 75% and 100% impacted, those differences, medians jump. We're finding a higher percentage of dead eggs in 75% impacted beaches and 100% impacted beaches, which shows us that the effect of the uplands can really affect egg mortality. And I do want to point out that impacted uplands has a lot to do with shading. If uplands has been impacted, a lot of the times those impacts include removing any tree cover that would be on those beaches. So like riprap, bulkheads, like a road, a lot of these like industrial additions can remove all those trees, which then remove shading off those beach. And as we saw in that previous graph, shading is really critical to egg mortality. So if there's no trees, then there's not going to be a higher amount of eggs that are dying. So in my opinion, impacted uplands and shading are very closely connected to each other, which is why we're seeing the significant differences between these medians in both the shading graph and in the impacted uplands graph.
All right, so moving on to the last variable that we've been kind of looking at. This is beach substrate. We'll start with a map and then kind of move on to some of the statistics. But first off, where are the type of beach substrates found that y'all kind of recorded on your field data sheets? These 2 maps, they display the types of beach substrates that are found across Fidalgo Bay, both from your sample scoring, which is on the left, and then on the right, this is a map that's from the 2001 shore zone mapping of intertidal substrate done by scientists and compiled across the region, actually. So overall, most of the substrate was mapped as medium and pea gravel across the bay sites, with a few sandy sites showing up in the surveys as well. The coarser resolution of the shore zone map to the right kind of shows the same thing, with the optimal habitat being targeted by you guys is 10 years worth of sampling. So good job kind of like working towards that optimal habitat that forage fish like look for spawning.
How does beach substrate affect egg mortality? So we'll be looking at 6 different types of substrates and their effect on egg mortality. So we have pure sand, pea gravel, medium gravel, coarse gravel, cobble, and riprap. And we'll start by looking at cobble and riprap. And like the shading graph and the fully exposed section from earlier, cobble and riprap have a very low sample size. And because of this, we can't say anything conclusive about the effect that cobble and riprap has on egg survivability. So until we collect more samples on cobble and riprap beaches, we cannot say anything about their effect on the percentage of dead eggs we're finding in any sample. So for right now, we'll look at pea gravel, pure sand, medium gravel, and coarse gravel. As you can see, our graph increases in sediment size from left to right, with pure sand having the smallest sediment size and coarse gravel having the largest sediment size. When we look at the medians, all of the medians for all of these beach substrates seem to be around 75%, with 50% of samples having percentages of dead eggs above that, from 75% to 100, and the other 50% being at 75% and below. And we're not seeing much change across the increase in substrate size. They all seem to be pretty similar. So the graph visually doesn't seem to indicate that there is a difference between egg mortality between these different substrate sizes. So let's look at statistics. I ran a Kruskal-Wallis rank sum test on this graph as well. And as you remember, we want a p-value that is less than 0.05. Our p-value is 0.5895, which means that there is not a significant differences between these means that can't be explained by randomness. So the slight differences between these means seems to be because of randomness and not because of some environmental effect that beach substrate has on the percentage of dead eggs in a sample. And this doesn't necessarily mean there is an effect on beach substrate on egg survivability. There just doesn't seem to be an effect that is represented in this graph. We would have to do further sampling and further graph making to see if we can pull out some kind of significant effect. But as of right now, we don't see any effect. Let's go over once again our 3 key points of this presentation. Number 1, of all the Department of Natural Resources aquatic reserves, Fidalgo Bay Aquatic Reserve has the most prolific spawning of surf smelt forage fish. As the years passed, surveys became more accurate and the number of eggs found increased. The changes in survivability of surf smelt eggs over the last 10 years does not show a clear trend. Egg survivability is influenced by seasonal changes and survey location. 3. Environmental effects, such as how much of a beach is shaded and the percentage of the uplands impacted by humans, have a statistically significant effect on egg survivability. And so let's look towards the future.
All right, looking towards the future, we look back on what you've accomplished over the past 10 years to inform how we move forward. So here's a map of the traditional sampling sites. You have space, generally they're spaced 100 feet apart from each other with your typical bi-monthly sampling strategy. So you can kind of see the map here kind of shows like the distances that you guys are covering with your sampling strategy. You have about 7,000 feet or around 2 kilometers of suitable habitat that you're actually sampling regularly. What you've done so far is randomize starting location and then 1,000 feet after that. So you're kind of like randomly subsetting or randomly like sampling these beach stretches every month. And this kind of like ended up being on the west side, you would have 4 to 6 samples, depending on the starting location, east side 2 to 3 samples, and then maybe up to 3 samples for special sites, which were kind of added mid-season and are kind of showing us some interesting trends, like a little different from just the general east and west side. Moving forward, we want to get the same data, but with fewer samples, kind of cut back on some of the work that's being done. And we're choosing to focus 1 site per month at each unique site, consisting of representative substrate or a shading impact or like an aspect. So kind of really directing science moving forward or monitoring moving forward to like be geared towards some of the questions you might have, which is, investigating like some of the effects on shading, or like maybe this kind of like aspect towards, like facing, due east as opposed to like northwest or something like that. Minimally, we recommend one side in the riprap on the north side, on the northwest side here, one side on the sandier southwest side, 1 to 2 sites on the east side, just because that is a bit of a larger area, and then paired sites on the spit. So the spit was kind of like, once again, that was one of the like sort of more special sites that we kind of like have looked at differently. One being a really shaded area, and then there's also a very like open area that could be paired, and you can kind of look at these direct effects of shading in that area. This would allow us to monitor the effect of upland impact in shading, as well as to parse out the microclimate effects of riprap and larger rocks. So we would have to do a little bit of thinking around classifying how we do our substrate and how we classify some of these things. But I think that microclimate question is a really good question to have. We also want to streamline the QA/QC process and reporting, and allow you guys to kind of pursue or collect data that allows you guys to pursue research that you might think are interesting. But overall, like, this is a good starting point and informed us really well of like some of the trends that we kind of thought were present, and 10 years of data is really valuable data to continue to like dig into. And then to move on after that, like focusing our research and monitoring on even more specific goals.
And just to finish off, thank you for the CSC for all the work that you've done over the last 10 years. And we're excited to work with you into the future and continue to collect samples at Fidalgo Bay, continue to monitor egg counts and egg survivability at the Bay. This is Leah and Ron signing off.